So, today we're going to be talking about nervous system cell types and anatomy. And we're first going to talk about the typical neuron that you find in most neuroscience textbooks. But actually, this typical neuron is the typical vertebrate myelinated neuron, okay? So, let's start with, we have typical neuron. But this is actually we're going to say, typical vertebrate myelinated neuron. Okay, so typical vertebrate myelinated neuron. All right, there's a lot of new words in there, so we're going to define all of them one by one. So vertebrate, that's any creature with a spine. So that's humans, dogs, cats, fish, so a lot of animals, okay? Myelinated, that's a new word. We're going to define that in just a second. And then neuron. So why am I making a point of all of this? So these typical vertebrate myelinated, motor neuron. Even more specific. Okay? Typical vertebrate myelinated motor neuron. All right. So, the reason why they're called motor neurons is because these are the neurons that attach to our muscles, and historically, they were always the easiest neurons to get to. So they're a lot easier to get to the neurons in your arm than it is to get to the neurons inside of your skull, in your brain. So these were the ones that were studied first. And so that's why in most neuroscience textbooks, you will see a typical neuron that is actually a typical vertebrate myelinated motor neuron. Okay? All right. So we are going to draw this typical vertebrate myelinated motor neuron in order to get some basic anatomy parts of the neuron. Okay? So let's draw this. All right. Well, actually, first, what is a neuron? Let's define a neuron first. Okay, so we have here neuron. Okay, so what is a neuron? Okay, so the word neuron, you might also see in the same place neuron with an E at the end. This is a very old-fashioned spelling. Or you might also see nerve cell. They're all the same thing. Okay? So, what defines a neuron? All right, so this kind of neuron, so what defines a neuron? Okay. So generally we talk about neurons as being able to receive, process, and transmit both electrical and chemical signals. Okay, so both electrical and chemical signals. And because they can interact with electrical signals, they are also defined as electrically excitable cells. Electrically excitable cells. This just means that if you can inject a current into a neuron or any cell that's electrically excitable, and if you inject the current, it will do stuff. It will get more active. Okay. Then, lastly, specifically our typical vertebrate myelinated motor neuron, it has what's called a soma, which is just a fancy Latin word for body. And the way you can tell which part of the cell is the soma or the body is because that's where the nucleus, it houses the nucleus plus organelles. Okay? So it has a soma. That's also a new word, so I'm going to underline it in red. And it has an axon and dendrites. Okay, more new words. So here we go. Axon, dendrites. Okay, so let's first define what these are. So soma, we already know, that's the body. That's where the cell keeps the nucleus and the organelles. And if you remember, the nucleus is where the DNA is stored. Okay? And organelles are like organs for cells. Okay? So, next, axon. What is an axon? Okay, so the axon is the part of the cell that carries electrical signal away from the soma, okay? Carries electrical signal away from cell body. And can you guess then what is the dendrites? Okay, so the dendrites carry electrical signal towards the cell body. Okay, all right, so now let's draw this. Now that we have some of our parts defined. Okay, so let's first draw the soma with some dendrites coming off of it. They look kind of like little monsters, little aliens. Okay, and one more, why not? Okay, and then we said it has a soma, dendrites, and also an axon. So the axon is very long, and then it ends in what's called the axon terminal. Okay. Axon is nice and long. All right. So here's a typical vertebrate myelinated motor neuron. So we're gonna give it its nucleus. That's how we know that that is the cell body. Then there will be some organelles in there. Maybe you recognize some of these structures from your biology class. Okay, so let's label some of these parts. So we've got here some dendrites. Okay, we've got here the soma. Okay, this is the nucleus. Nucleus. Okay, then this entire thing is the axon, and this is called the axon terminal. Okay, now we still haven't addressed the myelinated part of typical vertebrate myelinated motor neuron. So let's do that. So myelinated means that the axon is insulated by myelin sheaths. And so when I say insulated, you can kind of think of it like the insulation on a wire, okay? If you've been working with your Latte Panda kit, and there's some little wires in there, and they're pieces of metal with something wrapped around it to keep the signal inside of the wire, okay? So the similar idea, the myelin sheath wraps around the axon, but kind of in little chunks, okay? So that's why I'm now erasing here. Okay? And then I'm going to draw some myelin sheaths. So they look a little bit like, did you ever have this experience where when you're little and you're not very good at swimming, and maybe your parents gave you like floaties on the arms, you know, to help you float? That's kind of what they look like to me. So, okay, here we go. Dun, dun, dun. Okay. We're going to draw a lot of these. Didn't draw that long enough. Okay. And then like this. Okay, so these are myelin sheaths, and each one is created and maintained by a glial cell called a Schwann cell. So these are very small cells, and there are many of them on this axon, creating myelin sheaths and taking good care of them, making sure the myelin sheath is healthy and not damaged. Okay? So let's label these guys as well. So this is a myelin sheath. Okay? This is called a Schwann cell, and the gap in in between myelin sheaths also has a name, and this is called a node of Ranvier. It's just named after some guy named Ranvier. Okay, so let's define some of these new words that we've got. So let's go with, firstly, myelin sheath. Oh, no, sorry. All right, so we have here this new word, axon terminal. Okay, so the axon terminal is the end of the axon. All right, and this is also where neurotransmitters are stored. And so neurotransmitters are actually created here in the soma, okay? And then they travel all the way down to the axon terminal, and that's where they wait, where they are ready to be released by a nerve impulse. Okay, that's also a new word, new phrase, so I'll underline that. We'll get to that in just a second. Let's define our node of Ranvier. Okay, so these are gaps in the myelin sheath. I don't need to say the. Disaster. Okay. All right. Gaps in the myelin sheath. Okay. And this is where the axon membrane, okay, so this black line, where the axon membrane contains many ion channels, okay? So many, just to emphasize it, ion channels. And ion channels is a new word. So we're going to underline that and we will define it. Okay? So have we labeled everything? Yes, we have. Okay, so what we're going to do now is we are going to take a closer look at the node of Ranvier. Oh, actually, I should define myelin sheath first. Okay, let's do that. So myelin sheath, you've been waiting. All right, so this protects the axon, okay, and it prevents, what does it do? It prevents the electrical signal from leaking out of the axon. Prevents, okay, prevents electrical signal from leaking out of the axon, okay? So let's really quickly talk about its composition. So myelin sheath composition. Okay, so I'm going to draw 10 boxes. The middle is 5, so then 1, 2, 3, 4. 1, 2, 3, 4. Okay, so Myelin sheaths are made out of water, lipids or fat, and protein, okay? So, first one, water. Water is between 25 to 40% of a myelin sheath, okay? So, we're gonna do 25 to 40%. So 4 of the boxes have been filled. 25 to 40% water. Okay? Then it is between 60 to 75% lipids. So it's mostly fat. Okay? So we're going to go from 25, okay? So we're going to fill in this plus 1, 2, 3, plus the rest of it. Okay, so 60 to 75% lipids. All right, and then the last part is protein. Let's use purple. Okay, so protein is 15 to 25%. Okay, so now we're gonna go. So that was 10, 20, 25. So 15 to 25% protein. Okay? So that is the composition of myelin sheaths. That is what is protecting the axon and preventing electrical signal from leaking out of the axon, okay? All right, so really quickly, we're going to define Schwann cell. I'm going to use a different color just because we're starting to get a little busy. So Schwann cells are the most common glia. Here's a new word. Glia. In the peripheral nervous system, glia of the PNS, which stands for peripheral nervous system. And we're going to talk a lot more about glia later. Let's just say for now that glia are the unsung heroes of the nervous system. If neurons are like, the lead guitarist of the band, or maybe just, all neurons are like the band, they're like the rock stars, then the glia are like the stage manager, the lighting designer, the sound designer, the guy who makes sure that the venue exists, and the person taking all the tickets at the beginning, the janitor, the people selling snacks and drinks. So glia are really important, okay? And the only reason why we don't talk about them as much is because it took a lot longer for us to be able to study them. They're a lot harder to study, okay? All right, so glia. We will talk more about glia in a second. And let me just check if I have room. Okay, yeah. All right, let's keep going. And what we are going to do next is we're going to take a closer look at this node of Ranvier, and we're going to see what is going on in that node of Ranvier. So how shall we do this? I'm going to pick this one. Okay? And I'm going to bring this down here. And I'm going to blow it up. And draw it a lot bigger. Okay? So, we have on one side our myelin sheath, nice and big. Okay? Then we have in the middle our axon membrane. Then we have on the other side the next myelin sheath. Okay. All right. So now that we've zoomed in, this is what it looks like. I said earlier that this is the part of the axon membrane that contains many, many ion channels. So we are going to draw lots and lots of ion channels. Okay, here we go. So, ba-doom, 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 ba-da-da. Okay, so now in here, I'm drawing, like, it almost looks like two beans next to each other, okay? These are actually proteins, all right? So ion channels are actually proteins that are embedded in the membrane, okay? So now we're imagining that in here, this is the inside of the membrane, okay? So we have ion channels there, we have ion channels down here. They're just everywhere. There's tons and tons of ion channels in this part of the axon membrane. Okay. And even though the way we drew them makes it look like there's only 2 proteins per ion channel, we are now going to zoom in on the ion channels, okay? Let's see, where is the best place to do this? Let's just do it off to the side here. Okay, maybe I'll do this. All right. So let's see, what color to use? What color haven't I used around here? Let's use red. Okay, so I'm gonna pick an ion channel, okay? And now we're going to zoom in on that, and we're gonna rotate our view as if we're looking down onto the surface of the axon membrane now, okay? Okay, so this is what it looks like. So we've got not just 2, but 4 proteins, okay? And they kind of do this thing where they look a bit like a flower because there's 4 of them, okay? And then, so they're embedded, surrounded by the axon membrane. Okay, let me just make this a little more clear that this is all filled in. Okay, and then if you notice, there's a tiny hole. What color have I not used? Oh well. There's a tiny hole here in the middle. Okay, that tiny hole is the ion channel. It's like a hole or a pore or a tunnel where ions can go through from the outside of the cell membrane. So in this case, from the outside of the axon to the inside of the axon, or from the inside to the outside. Okay, so let's define our terms here. Let me just rewrite it someplace where I'm not going to need to use the space. Okay, so over here. Actually, I wonder if I can just fit it up here. Okay, so what is an ion channel? An ion channel is proteins in the membrane, okay, that create a hole for ions to pass through. Okay, so the next question is, we have another new word here. What is an ion? Okay, so an ion, I'm running out of colors. We'll just stick with blue. So what is an ion? An ion is, okay, so an ion is an atom or a molecule that has an uneven number of electrons or protons. So if you think back to your biology or your organic chemistry, electrons and protons are fundamental parts. They're like particles of atoms and molecules, and they carry charge, okay? So if you have an even number of electrons, which are negative, and protons, which are positive, then the charges all cancel out, and the molecule or the atom itself is neutral in charge. But an ion is an atom or a molecule that doesn't have an equal number. So it's either positive or negative, because it either has more protons if it's positive, or it has more electrons if it's negative, okay? So an ion is an atom or molecule that carries, so with a positive or negative charge. All right? And then the last thing that we're going to do in this video is we are going to take a closer look at what goes on in the part of the axon membrane that doesn't have an ion channel, okay? So we'll just do this. So we're going to zoom in on the membrane itself, and the way that this is formed, the structure, is there's a bunch of heads with tails. They look kind of like tadpoles, like mutant tadpoles with two tails, okay? And then on the other side is another layer. But if you'll notice, all the heads are facing outwards, and all the tails are facing inward, okay? So this, if we zoom in, this black line becomes all of this. So what is this? Okay? So, okay, so this is what is called a hydrophilic head group. Okay? Hydrophilic means water loving. Okay? And then the tails, these are hydrophobic lipids. Hydrophobic lipid tails. Okay? In most animal cells, this hydrophilic head group, head group is the very generic term, but in animals it's usually a phosphate. Okay? Which is why this whole thing is called the phospholipid bilayer. And you've probably heard that term in a biology class at some point. Okay? All right, so this is our typical vertebrate myelinated motor neuron. Historically, one of the first neurons that we studied. But in the next video, I'm going to talk about the 5 major categories of neurons based on their structure. And this is only one of them.